Hi, my name is Rohan Patel, and my project this year is about the heterogeneity in excitatory and inhibitory GABA responses in adult MCH neurons. Last year, I worked on a project involving techniques to detect amino acids for a neurological effect from a disorder known as vasospasm. And the field of neuroscience has never failed to intrigue me. This year, I'm working with a new mentor that incorporates electrophysiology in the study of the hypothalamus. After last year's project, I wondered about the brain's response to different acids that are present in certain parts of the brain. I talked to my mentor from last year about working on a project similar to what I had in mind, and he arranged me to work in the lab with one of his colleagues. The project will be taking place at the Yale Department of Neurosurgery, where I will be working alongside with my mentor, Dr. Daniel Spergel, who will be responsible for obtaining the brain slices and recording area and action potentials of the MCH neurons. Dr. Yang will be injecting the mice with expressions so we have the ability to clearly identify the cells needed for analyzation. I am part of the pre-lab prep, making the ACSF and the cutting solution to keep the brain slices in while searching each slice for a healthy cell. I'll also be observing the recording of the area and action potentials of the MCH cells. To understand more of the project, let's go over some important background. MCH neurons, abbreviated for melanin concentrating hormone, form a unique group of cells in the hypothalamus with axonal projections throughout the brain, which play a critical role in sleep and energy homeostasis. These neurons are predominantly found in the parafornical area of the hypothalamus. MCH neurons express GABA A receptors, and GABA accounts for the majority of synaptic input to MCH neurons. Stimulating MCH promotes sleep, but most sleep-promoting drugs used clinically are inhibitory by activating GABA A receptors. So GABA promotes sleep by inhibiting HCRT neurons or hypercretin, which is important for exciting MCH cells. GABA and glutamate will be compared during the experiment, and antagonists for each will be used as well. Bicukulene blocks GABA and AP5 blocks glutamate, and these allow the other acids' responses to be solely measured. There are GABA-B and GABA-C receptors, but these are not found in MCH neurons. The main concept to know for the experiment is that glutamate excites and GABA inhibits. However, there's a possibility that GABA excites MCH neurons. To analyze the obtained brain slices, we're using an Olympus microscope to measure the area of the neuron. An electrical stimulator is used to measure the action potentials of the MCH neurons. Mice are injected with a channel rhodopsin, TD tomato, or GFP expression for study under the microscope. Slices are cut at intervals of 300 micrometers and transferred to the microscope, which has ACSF running at all times to keep the slice alive. We use either blue light for GFP slices or green light for channel rhodopsin to find a cell. We either measure the action potential of the whole cell, or we use a gramocydin solution placed in the action pipette that is part of the electrical stimulator and perforates the cell so we can see the exciter or inhibitory response. Antagonists must be utilized because both glutamate and GABA are present in the cell, so only one is solely measured. The bursts of both glutamate and GABA are then compared and checked for differences. Thank you for watching my video.